Damage to the central nervous system, such as occurs in spinal injuries, doesn't heal, and deprived of nervous connections to the brain, large parts of the body are paralyzed. Until quite recently, medical care concentrated on helping the patient come to terms with their permanent disability. But today, patients with spinal injuries are encouraged to keep their bodies in trim, since the possibility of stimulating the regeneration of central nerve connections seems to be just around the corner. And research indicates that successful regeneration will require nerve interaction with healthy tissues. We've come to the University of California's Developmental Biology Center at Irvine to look at examples of the fundamental research on nerve development and regeneration on which such exciting predictions are based. But all the research carried out here is on animals rather than humans. Why?
The main reason for working on animals is the stages we care about are the earliest stages of development of the nervous system. That's when the most critical events are taking place, and those events in mammalian species, such as man, are happening inside the womb, where they're inaccessible and impossible to observe without going to heroic measures. We're interested in these early events because it's those early events that set up all the later development of the nervous system. And those are interesting from several different viewpoints. Basically, it's an intrinsically interesting issue, how the body develops, how the nervous system develops. But not only that, by knowing how it develops, we have an idea of how it can go wrong, and hence have some understanding of birth defects. Also, by knowing how it develops, we are hoping to understand how we might make it redevelop or regenerate, and hence have implications to the medical field.
In my research, I use chicken and quail eggs because development of birds, particularly in the early stages, is quite similar to human development. Unlike mammals, which develop in utero, birds develop in an egg, which is easily accessible to experimental manipulation. One can open up the egg, perform an operation, close it back up, and the embryo will develop perfectly normally.
An additional advantage of using bird embryos is that there are cell markers available. For example, chicken and quail both develop at similar rates, and it's quite easy to graft pieces of quail embryo into a chick embryo. And because the cells of the two species are readily distinguished, it's possible to follow the progeny of the quail cells during stages of the chick's development. In birds, the nervous system develops from a flat sheet of similar cells called the neural plate. This plate then folds to form the neural tube. In the head region, the neural tube expands to form the brain, and in the trunk region forms the spinal cord. The entire central nervous system is composed of the brain and the spinal cord. In addition to the central nervous system, there is also the peripheral nervous system. The peripheral nervous system arises from a group of cells within the neural tube called the neural crest cells. These cells migrate away from the neural tube to form peripheral ganglia, which send out axons that connect back to the central nervous system, relaying information from the peripheral nervous system to the brain. In this process of development, the similar cells of the neural plate form the cells of the central nervous system, and, migrating as neural crest cells, form the different neural and glial cell types of the peripheral nervous system. So development is accompanied by changes in cell type and cell position.
There may be a functional significance other than just the position of the cell bodies between the peripheral versus the central nervous system. For example, in regeneration, an injury to a peripheral neuron, say a sensory nerve in the hand, would be expected to lead to a period of numbness following recovery of function and feeling in the skin when the nerve regrows. A similar injury in severity to a spinal cord would lead to loss of function and no regeneration. Now, one of the few exceptions to that is in some animals, the optic nerve seems to be able to regenerate. For example, in frogs and other lower vertebrates, following an injury, the nerve can regrow and over a short period of time reestablish connections. And the hope is that the regeneration seen there may relate back to man, since early in the development of mammalian species, it appears that there may be similar regenerative abilities.
The nervous system is a very complex structure composed of many different types of nerve cells that are intricately interconnected. A general question that we're interested in studying is how the cells of the developing nervous system know how to get to the right places and at the right times. This question is so complex that we go to a simpler system to explore it, and the system that I've been using is the neural crest. Neural crest cells migrate extensively in the embryo to give rise to the peripheral nervous system. We use a variety of techniques to study the patterns of neural crest cell migration and to test the possible function of molecules that may be involved in guiding their migration. A first important step is to establish exactly how the cells migrate, and in order to do that, it's necessary to mark the cells in some way.
As cells multiply and migrate, differentiation takes place. And while there may be no visible difference between the cell types, they may be labeled by adding fluorescent antibodies that bind to specific molecules on the surface of the target cells. A second method is to perform grafting experiments using visibly identifiable marker cells from one species and introducing them into another. A third method is by injecting a dye that labels the target cell and its descendants. This last method, the injection of a fluorescent dye into the neural tube, enabled the researchers to follow the neural crest cell's migration in forming the peripheral nervous system.
What we find is that the peripheral nervous system that's derived from the neural crest is highly segmented. We can get some clues about how this segmentation might occur by looking at the early embryo. Adjacent to the neural tube are condensations of mesoderm called somites, and these are segmented in precisely the same pattern as neural crest cells. If we look at the pattern of neural crest cell migration through the somites, what we find is that they migrate through only one half of the somite and not through the other. And we can find this by staining the cells with antibodies that recognize the surface of the cells. What happens is the neural crest cells migrate through one half, and the peripheral ganglia that are derived from the neural crest also occur in the same half of the somite. So what we see is that this pattern of migration is highly segmented and seems to be related to the segmentation of the somites. Establishing the pattern of cell migration is an important first step in identifying how cells actually migrate, but it tells you little about what the mechanisms of migration are. The second step is to try to identify candidate molecules which may be involved in the migratory process. My laboratory has particularly concentrated on molecules that may be present on the surface of somite cells or in the extracellular matrix. We look at the distribution of these molecules using antibodies. And to date, we found a molecule that's distributed in the opposite half of the somite to which neural crest cells migrate. This makes it a good candidate for playing some kind of guiding role in neural crest migration. However, simply by looking at the distribution, we really don't know what the function of that molecule is.
The question that my research centers on is how cells become unique, both in cell type and in position. If we move to the eye, for example, there are different cell types that make up the neural retina so that it functions as a working organ. How do those different cell types arise? Not only that, how do the cells in different regions of the retina gain some positional cue so that they can then connect properly to the brain? To ask questions of this sort, it means we have to work on organisms that are accessible. Chicken would be one way to go, but it's wrapped up inside of an eggshell. So most of our work involves the frog embryo. So that means that the events in early neurogenesis that we most want to study, we can directly observe. In addition, the development happens very rapidly. We move from a fertilized egg to an animal with a working visual system in a matter of days. Finally, the cells are very large, and the animals are very hardy. That means that we can do cut and paste experiments, grafting cells from one organism to another to challenge any mechanism that we might propose as important for neural development. Now, in considering uniqueness of the cells, uniqueness cell type or position, It's always boils down to a question of nature versus nurture. Now, Sidney Brenner put the question best, I think, and when he proposed that the question is it that neural development proceeds on an American plan or a European plan? In his proposal that by the American plan, the cells would develop according to their neighbors. In the European plan, instead of developing according to their neighbors, they develop according to their ancestry. Well, to ask questions about ancestry means we need to have some way of following the family tree of the cell. In the case of the cells, it's called the cell lineage. Now, in some invertebrates, it's possible to directly observe the cells as they develop, and in that way find out which cells and which cell types descended from which precursor cells. In vertebrates, that fails miserably because there are too many cells and they look too much alike. So to make them unique, what we do is go in and inject a single precursor cell with a fluorescent dye. This fluorescent dye then can be followed as development proceeds, and we can determine who the great-great-great-grandchildren of the injected precursor are. Now, in doing this, we can ask very basic questions. For example, when does the central nervous system and the peripheral nervous system diverge? That question would be easily asked by injecting a cell and finding out if the great-great-grandchildren contain both central and peripheral nervous system cells. And in fact, in most of our clones, we find that that does happen. What that means is that there must be a common ancestor between the two, and it's looking very much now like it's location that dictates whether you become peripheral or central nervous system. That is, that choice is made by the American plan. Well, to assess the importance of cell lineage and the assignment of cell type, we can inject a single precursor cell with the marker dye and follow what its great-great-grandchildren are as far as their cell types. To date, we've addressed this question in the retina and in the spinal cord of chickens and frogs and in the neural crest of chickens.
All the precursor cells are identical in appearance, but a developed organ, such as the eye, consists of several different cell types. According to the European plan, cell type is determined by ancestry. If we label one cell, then at any intermediate stage of development, all its progeny will be destined to become the same type of cell. All that is necessary is that the cells find their way to positions appropriate to their predetermined cell type. According to the American plan, the progeny do not inherit a predetermined cell type from the labeled precursor cell. But at some intermediate stage in development, they become sensitive to positional cues from their neighbors. And it's these positional cues that determine the type of cell that the progeny will become.
And what we find is that there's no predictable relationship between the great-grandchildren. Therefore, it looks like cell type is dictated by the American plan. That is, local interactions telling the cells what to become rather than their ancestry. To ask questions about positional cues in the formation of neural patterns, we have to first ask if the fibers make unique patterns as the projection from the eye to the brain first forms.
Using the technique of confocal microscopy, it's possible to follow the growth of the optic nerve axons in living frog embryos. Here, time-lapse film of an optic nerve grown in culture shows how the axon growth cone with its filopodial arms seeks to find the route to the brain. While such observations can give information about the cues that guide the migrating axon, to determine the pattern of connections between eye and brain, cell markers are needed. Different color dyes can be injected to mark different groups of cells, even individual cells, within the eye. After development of the optic nerve, the appearance of these colors in the cells of the brain enables the pattern of connections to be mapped. Let's first consider what such connections must do. The image projected by the lens on the inner surface of the eye, the retina, is upside down and back to front. The connections to the brain unscramble these reversals. So a labeled neuron at the front of the retina develops an axon connecting to the back of the brain, one in the bottom to the top, and so on.
Given that they do show unique patterns, we can ask if they get the positional cues that guide this patterning based on an American plan or a European plan. We can do this by grafting cells from their normal neighborhood to an abnormal neighborhood. Obviously, if the American plan matters, the cells should now act as their new neighbors. If the European plan is the important aspect of this development, they should act according to their ancestors, independent of their neighbors. The results have been very uniform. The cells always act according to their ancestry and never according to the neighborhood to which they're grafted. So to summarize, what we have is two different aspects of neural development that appear to use somewhat different rules. The cell type that cells develop into seems to be dictated by an American plan. The neighborhood matters a great bit, whereas the positional cues that underlie the assembly of neural connections seems to be mostly a European plan. That is, the fibers seem to choose targets dependent on their ancestors. Well, obviously our knowledge is still far from complete, but what we do know so far says very strongly that there are multiple interactions or cues guiding the fibers in their selections of targets in the brain. To test whether or not these are sufficient to explain the patterning that we observe, we can use computer simulations in which we model thousands of optic nerve fibers innervating the target tissue. What we find, even starting from a random starting condition, is that these fibers slowly sort out their correct target site using this combination of cues. Most important for our purposes here is that we find that there's a great robustness in this system. We can eliminate individual cues and the fibers will still find their normal target sites, although perhaps a little bit less quickly and perhaps a little bit less accurately. Nonetheless, a largely normal pattern can emerge.
So far, we have looked at the factors that control the development of nerves in the embryo. Dr. Ron Meyer's studies concentrate on the optic nerve in adult animals.
My research is aimed at trying to understand how axons in adult animals respond to nerve injury. And we study two animals. One is the goldfish, and the other one is the mouse. We study the goldfish because it has a remarkable property. It has the capacity to regrow nerve connections that are severed.
What is done experimentally is to sever the optic nerve. The part that leads to the brain then dies because it's separated from its source of nutrition. The part that's still attached to the eye lives on because it's still attached to the cell body which resides in the retina. These severed axons form new growth processes at the point where they're cut, and these ends, with their filopodial arms, which look very much like the growth cones that are seen in developing axons, regrow back into the brain and reform a very precise set of connections. The very precise nature of these connections can be measured with exquisite detail. By carefully positioning an electrode in the optic tectum of the fish's brain, it's possible to record from individual optic nerve fibers and correlate their signals with specific points in the fish's visual field. And then, as regeneration occurs, determine the pattern in which the nerve fibers reconnect to the brain.
Basically, what we find is that given enough time, and this is on the order of a few months, those connections are almost as good as a normal animal. How does a nerve fiber in an adult animal do this? Do they use the same kind of cues that a fiber which is growing in an embryo might use?
Ron Meyer came to the conclusion that regenerating fibers in an adult goldfish use two of the same cues that developing fibers use to establish an appropriate set of connections to the brain. One of those cues is a local chemical address system. Fibers have the capacity, as they're growing, to read the local cellular environment and then navigate according to cues that the growth cone sees. And they've done a number of experiments to show that is what is used by regenerating fibers in the adult as well. For example, when they tried to make fibers grow from a wrong place, the fibers recognize this wrong place and alter their course so they get back to the right place. This gives them a rough ballpark set of appropriate connections.
The second cue that fibers use is an activity-dependent fine-tuning process. It turns out that even in the dark, there is a continuous barrage of activity that is propagated from the retina back into the brain. And it's an orderly activity such that local regions of the retina tend to fire at the same time. There's a slight tendency for them to do this, and the fibers can recognize this tendency to fire, that they fire at the same time, and recognize who their neighbors ought to be, and they adjust their connections so that those fibers that are firing together come to be grouped into one spot. And that gives you a very precise local order. So the first cue of this chemical navigation gives you a ballpark or global order, while the second cue fine-tunes this order to give you a precise local order. Well, we also work on the mouse. And mouse is very different than a goldfish. A mouse is like us and other mammals. When the optic nerve is damaged in the mouse, it doesn't regenerate. And after a while, the cells eventually die. This is rather similar to the case of spinal cord injury, where the nerves that come down from the cerebral cortex into the spinal cord also are unable to regenerate once they're severed. Now, recently, however, we have found that we can take pieces of the retina out of the mouse and put them into a culture dish and keep them alive. And under certain circumstances, we find that these pieces of retina are able to grow new nerve connections. We see growing axons coming out of these little pieces of retina. So what that tells us is that the nerves have the capacity to grow, but there's something about being inside of the animal, inside of the central nervous system, that is preventing these nerve fibers from growing. And more recently, what we've found is that when we damage the nerve, these nerve fibers begin to synthesize a protein called growth-associated protein, which is normally found only during embryonic growth. So it's like these fibers are really trying to grow. They have all of the machinery for growing, but there's something that interferes with that growth. Now, we think that if we can get them to grow, from the work that we've done in the goldfish, that they would know where to go, because the cues that seem to be there are in the adult brain in goldfish, and presumably also in mouse, are very stable cues. So we would anticipate that once we can get them, if we can get them to grow, then they would automatically grow to the right place in the brain and form an appropriate set of connections to mediate visual behavior again.